A professor once told me when I was trying to interpret a scientific figure that this is genuinely scientists trying as hard as possible to make things understandable. But biological data visualizations can often be very difficult to interpret, so here I am to explain a very common one called a volcano plot. A volcano plot is used to interpret RNA sequencing data, and what it allows us to do is identify genes that demonstrate strong and meaningful changes in gene expression between two conditions. So on this plot, every point represents a gene, and the x-axis shows us the log2-fold change, which shows us how much a gene increases or decreases in expression between those two conditions. So genes that are far to the right and positive on the x-axis show genes that are increasing in expression, in this case our tumor condition on this plot. And genes that are far to the left and are negative on the x-axis show genes that decrease in expression in the tumor condition relative to the normal condition. The y-axis shows us statistical significance. So I think that this can be confusing because normally when we are studying statistical significance, we may be looking at the p-value, and in that case, we want the p-value to be as small as possible. Taking the negative log of the adjusted p-value is just a statistical transformation that we do so that the greater the value, the higher the statistical significance. So on this plot, genes that are further to the edges and higher up are genes that demonstrate strong and statistically significant changes in gene expression between the two conditions, and are therefore genes that we are more interested in. This plot is practically useful in myriad ways, one of which is that these interesting genes along the edges and far up on the plot are ones that may be drivers of the pathology that we're looking at. So in this case, we're looking at a tumor condition versus a normal condition. So these interesting genes could be driving the pathology that causes that tumor, and are therefore also potential druggable candidates. It's unlikely that we'd want to look at these genes that are kind of close to the middle and not really, statistically significant on the plot, because it's unlikely that they are contributing to the pathology there. Thank you so much for watching, and I'll see you in the next video. Bye.